is a seasoned professional in the field of seed technology with 12 years of experience as a technical manager at the M&T Seed Testing Laboratory, the first ISTA accredited laboratory in Bangladesh. He has been actively involved in various aspects of seed quality control, quality assurance, and quality management system. His expertise lies in ensuring adherence to international standards and implementing quality control measures to maintain the highest standard in seed testing and production. Now, with that introduction, may I call upon Mr. Azizul to wrap up and for the closing remarks. Thank you very much, Mr. Azizul, all yours.
Thank you, Dr. Uma, for giving me the opportunity to deliver the wrap-up and closing remarks in this webinar. Hello, everyone. Good morning, good afternoon, and good evening, wherever you are joining from. Standing Committee on Seed Technology, representing Lalti Seed Limited, Bangladesh. First and foremost, I would like to extend my sincere thanks to all the active participants for joining this webinar and engaging with such attention and interest. A heartfelt thanks to Dr. Uma for skillfully moderating the session and to Dr. Tomoko Sakata for wonderful opening of this webinar. I would also like to express my gratitude to the chair, co-chair, all the respected members to the Seed Technology Committee, and of course, to the entire AFSA team for their excellent coordination and support. Above all, my deepest appreciation goes to the distinguished speakers whose insightful and informative presentations truly made this webinar meaningful and impactful. Nowadays, modern agriculture faces significant challenges, from the effects of climate change and the decline in soil fertility to the overuse of chemical inputs. Today's webinar is organized around solving these problems through sustainable seed enhancement, and the speakers of this webinar have tried to show ways to solve these problems. I will now provide a brief overview of the key points raised by our speakers. A very special thanks goes to Dr. Helena Freitas from the University of Coimbra, Portugal, for her powerful and eye-opening presentation on the role and benefit of seeds and seed technology in regenerative agriculture. In her talk, she shared some very important facts. By 2030, 575 million people will still be living in the extreme poverty. At the same time, our food systems are causing serious harm. Over 1/3 of greenhouse gas emissions, up to 70% of freshwater use, and 80% of biodiversity loss come from how we grow and produce food. She explained that a sustainable food system must provide food for all now and in the future, while also protecting the environment and supporting society. Then she introduced the One Health approach. This idea shows how the health of humans, animals, plants, and ecosystems are all deeply connected. Protecting one means protecting them all. She also spoke about the role of the soil microbiome, tiny living organism in the soil that helps plant, support ecosystems, and improve human and animal health. These invisible allies are key to restoring our soils. Through agroecology, she gives us a wonderful path forward. Agroecology is more than farming. It's a science, a set of practices, and a movement that values nature, local knowledge, and fair food systems. At the center of all this, she placed the seed. She told us they are diverse, locally adapted, and climate resilient seeds are essential for the future. These seeds help crops survive drought, heat, floods, and other challenges. She also showed how seeds can carry beneficial microbes, helping plants grow better and reducing the need for chemical fertilizers. She acknowledged that spreading these microbiomes across large farms is expensive and not practical yet, but solutions like seed coating, adding beneficial materials directly to the seed can help solve this. In the end, her message was clear: the seed is small but powerful. It carries the potential for healthier soils, stronger farms, and a more sustainable future for everyone. I would like to take a moment to reflect on the valuable insights and shared by our second session speakers, Barry Hoff and Carola Peters, lead research scientists from Incotech, on the topic biological applications on seed for regenerative agriculture. Barry stated that agriculture is undergoing a shift, especially in EME regions where farmers are moving toward microplastic-free, biodegradable solutions, as well as practices like vertical farming, hydroponics, and polyculture. These trends reflect a growing need for sustainable and environmentally responsible approaches. He explained that this change is largely due to rising challenges such as climate change, soil salinization, and tighter legislation which are pushing growers away from conventional farming practices. To address these challenges, Barry Hoff presented the importance of understanding how plants respond to abiotic stress. He emphasized that a critical first step is to observe how plants react to stress conditions like drought and salinity, and to measure indicators such as reactive oxygen species. As he showed through the tomato seed trials, timing and measurement matter significantly when evaluating stress responses and developing effective seed treatments. He showed that reactive oxygen species levels can help us identify how stressed a plant is and guide the development of seed treatments that improve plant resilience. Next, Carola Peters discussed the role of biologicals, a wide range of natural inputs like plant extracts, amino acids, and beneficial microorganisms. She stated that microorganisms are particularly effective because they colonize the root zone early, helping plants absorb nutrients and outcompete harmful organisms. She presented research on how different coating technologies can deliver these microorganisms. However, she pointed out that compatibility is crucial. Microorganisms must be able to survive alongside film coatings and fungicides, whether chemical or biological. Carola showed example of compatibility testing between coating and microbes, and also emphasized that not all microbes work with all crops. For example, soybeans require specific strains of rhizobium, while chickpeas and lentils need others. She explained the importance of timed release from the seed coating, ensuring that the biologicals are active just as the seed begins to grow. Finally, Carola and Barry emphasized a clear message: each crop faces unique challenges, and biological seed treatments must be targeted and compatible to be effective. By applying the right solutions, farmers can reduce chemicals use, increase crop resilience, and contribute to a more regenerative, sustainable agriculture system. In the end, uh, if we want a better future for farming, we need to use smarter and safer methods. Sustainable seed enhancement is one of those methods. It helps plants grow better, protects our soil and environment, and reduce the need for harmful chemicals. By working together, farmers, scientists, and decision makers, we can build a healthy, strong, and sustainable farming system and ensure food security for all. Thank you again to our speakers and to all of you for being a part of today's webinar. Thank you.
Thank you very much, Mr. Azizu, for the closing remarks. So, Before we finish the webinar, I just would like to share with everyone about our seed technology webinar next session. Session 3 will be on the seed diagnostic technologies, and it will be held on July 24th. So we still have 2 months to go for the next webinar for seed technology. And for those who are about to leave, you can scan the QR code here to fill up the post-webinar survey. Once you fill it up, you get an automatic e-certificate of attendance after you complete the survey. The presentation and recordings will be shared on APSA website, and our team, Bobby, will send an email notification to you, including the link of the post survey. So with that, thank you very much, everyone. Take good care.
All right, ladies and gentlemen, and with that, we will have a short 30 minutes break. We're actually on time. So hopefully we can start at exactly 4:30 for our final session of the day. Right? Thank you very much. Oh, and refreshment has already outside. So feel free to help yourself. There are packed food and there are hot drinks. So please help yourself because yesterday there's a lot of extras. So hopefully today everyone can take some. All right. Or if you're not eating here, you can also bring that back. Right? Okay. Thank you. Yes, Assalamualaikum and good afternoon, everyone. I believe we can get started, right, so that we can finish on time. All right, so ladies and gentlemen, the theme for our next segment is seed industry development in Malaysia. And this segment will be moderated by Professor Dr. Umarani Sinia, President, National Seed Association, Malaysia. And allow me to introduce on Prof. Umar. All right, so Prof. Umar completed her PhD in 1997 from the University of Reading, United Kingdom. Her field of expertise is seed science and technology, covering physiology, biochemistry, and storage for both conventional and via cryopreservation. And currently, she is holding the post as a Deputy Dean, Graduate Studies, Research and International at Faculty of Agriculture, UPM. And without further ado, I would like to invite Professor Dr. Umarani to moderate our next session.
Thank you very much, Dr. Shazwan, for that introduction. Hi, everyone. No energy? No energy left? Come on. Look, I'm so energetic, so better all of you also join me and be energetic. Okay, so this afternoon, or rather evening, we have got 3 more speakers. Very interesting talks, and mostly about breeding, right? I think you all have been hearing about breeding earlier on. So this is going to talk to us about the conventional breeding method, what are the new breeding methods, and so on. So I would like to invite the 3 speakers. Dr. Shima, right? You can sit on the VIP chair in front. Emilia, Miss Emilia, come. And then the 3rd speaker is Dr. Muhammad Shaifuddin, right? So, okay, so our first speaker will be Dr. Nazatul Shima, and her is plant breeding and genetics. Okay, so she actually got a bachelor's in chemistry and then MSc in plant science from Wisconsin Madison, USA, and her PhD from Wageningen University, or the other way around. Okay, your bachelor's was from USA, your MSc was from Wageningen, sorry, and PhD was from UPM. Okay, my apologies. Too excited. Dr. Shima was appointed as tutor from 2007 to 2017, during which she pursued her master's and PhD, worked in maize breeding projects, and currently she is not just working with sweet corn, grain maize, forage maize, hybrids, inbred lines, but she's also working with many other potential new crops like sorghum. Developing plant tolerance to abiotic stresses via conventional breeding, marker-assisted selection, and polyploidization approaches. So with that, I would like Shima to deliver her presentation entitled Breeding for Food Security. That's all I can read. Please welcome Dr. Shima.
Hello. Okay. Thank you very much, Prof. Umar, for that kind introduction. Assalamualaikum warahmatullah, and very good evening. Thank you for having me in this auspicious event of S.E.E.D.S Symposium. I'm very honored to be here to deliver my talk on breeding for food security, cultivating resilience in a changing world. So a very fancy topic. stories of our breeding efforts done in UPM in tackling challenges for climate change, basically. Next. So the content of my topic my talk will be on some general introduction on food security and the meaning of resilience in crop. And then I focus on the role of plant breeding in combating or tackling food security. And then I will focus more on the third content on the breeding efforts done in UPM in tackling climate change, in developing crops, climate-ready crops. And finally, I summarize with stating some future direction in breeding for food security. Next. First, I will start on the current status of food security. Next. So this is a map from FAO, just showing the prevalence of food insecurity into 2020 to 2022. So you can see that just focusing on our country here, Malaysia, we are not doing that bad, actually. Comparing, we are 39th out of 113 countries based on the Global Food Security Index. In 2021, we are ranked 8th among Asia Pacific and 2nd among Southeast Asia after Singapore. So what is actually food security? What are the elements or what are the components that are visualized or defined food security? So these are the 4 components of food security. In order to be food secure, the components of availability. So food must be, there are a reliable supply of food and insufficient amount and quality. So it must always be available for our people. And the next component is on utilization. And the food is not only available, but there must also be nutritious and adequately utilized by body. So good food, nutritious food. Next component is stability. And the food must be permanent and durable. We have permanent and durable access to food. It must always be available and stable. The production is stable across whatever challenges and condition of weather. And finally, accessibility of food. Individuals and households have adequate resource to obtain appropriate food. So these are the 4 components of food security, but I'm focusing for breeding specifically and agriculture in general. We focus on these 3 components that can be catered directly in the agriculture field. The availability of food, cultivating or breeding for nutritious food, and cultivating for food that are stable, stable production across harsh environments. Next. So the breeding program or research strategy in UPM focus on following this strategic plan, plan tindakan dasar security makanan negara DSMN, which have listed all these 5 core strategy and focusing on strategy TS or Teras Strategik kedua, permekasaan, kajian dan penyelidikan, research and development. So under that core strategy, there are 2 strategies. The first one to develop crops or varieties that have high yielding and high resilience. And the second strategy is to identify alternative potential crops. So in line with this core strategic number 2, and in line also with the activities listed under the 2 strategies of research and development to ensure food security, I highlighted 3 activities in this document that where we plan our breeding programs and our research activity in UPM. So for my topic or my talk further will emphasize on these 3 activities in developing varieties, crop varieties that are high yielding and tolerance to biotic and abiotic stresses, and climate ready crops. And also the blue one actually corresponds to the second strategy, on developing or exploring new alternative crops for food as well as feed for animal consumption. Next. Resilience. When we talk about resilience, we are talking about how to breed or select or develop crop varieties that are resilient to climate change. The perfect storm here describe a situation where we are moving towards that. Is happening now with increasing population and increasing level of urbanization, with increase or induce what we are experiencing of climate change. The resilience in agriculture refer to the capacity of our agriculture system to adapt or withstand any changes in environment while maintaining productivity, stability, and ecosystem health. So sustainability, sustainable agriculture. So in order for us to make sure that we have resilience in agriculture, breeding plays an important role to start or to kickstart any the agriculture ecosystem. Then how the resilience as a response to climate change describe on how we enhance this resilience in food system to mitigate the impact of climate change and ultimately to ensure that food security are achievable through adaptive strategies, mainly focusing on breeding programs. Next. These are some of the effects of climate change. I believe most of us are already well aware and already experiencing the increased temperatures, increment of temperatures, and as you can see in the this side, this graph shows the effect of every 1 degree Celsius of increase in temperature. How does it affect the change in yield? So as the bar goes to the that side, it's actually a yield reduction. So we are already aware that the detrimental effect of temperature increase effects in plant performance. Next. Other than that, we are also experiencing increase of sea level because of the melting of the ice caps. As you look at the map here, our country is prone to flooding risks. So these are some of the challenges that we are currently facing and will be facing in the future. So we have to make some move towards ensuring food security by plant breeding approaches. So the role of plant breeding in ensuring food security started with some of the history. Of course, plant breeding is one of the earlier art, I would say, in agriculture, where breeders have this well identify potential crops, and then by selection or selective breeding, we choose parents with desirable traits to produce offspring that have favorable characteristics. So these are some of the advances in traditional plant breeding or conventional plant breeding, where it started with using in corn, rice, and soybean, of using open pollinated varieties, and then SHAH and as well as Norman Borlaug and this Professor Dr. Yuan Longpin, some of the important figures in plant breeding, developing a hybrid breeding method that can improve general agronomic yield. So breeding, conventional breeding has of course proven it a good track record in improving yield generally. Next. But now with the advance of modern biotechnological approach, we have marker-assisted breeding as well. The use of DNA marker that can accelerate breeding program, okay, by using molecular markers as selection tools, together with the phenotypic selection, of course. So the conventional or the phenotypic selection as well as marker-assisted method marker-assisted breeding must go hand in hand because we have to validate the performance of this crop on the field as well. So, and then we have up-and-coming technologies that are applicable for genome editing and CRISPR-Cas technology to edit genes of interest that have identified to for tolerance against abiotic stresses, and this can increase the precision as well as to expedite the breeding process to introduce beneficial traits to enhance food security. So these are generally breeding, how breeding can increase or contribute towards producing varieties that are adapted to climate change. Next. This is the focus of my talk today, how we in UPM cultivate resilience to breeding effort. I will just go through all of the breeding programs that have taken into some of the breeding projects that we have already conducted in UPM, and of course, some of the projects are still ongoing. I believe everybody knows about Padi U Putra. Padi U Putra is one of the program that has started in early 2010 until 2016 under receiving the long-term research grant. So the focus of this has been realized by, of course, by the ministry at ministry level, and then UPM has taken this and produced a variety to in extensive breeding program, producing Padi U Putra, which has resistance against blast. So climate change is not only about abiotic stress; it also produces emergence of new diseases. So that's why breeding for biotic stresses is also one of the breeding approach done, breeding program done in UPM. So Padi U Putra is produced by using marker-assisted back crossing breeding. So conventionally, a back crossing method would take about 5 to 6 generations of back crossing, and by using marker-assisted back crossing, using the particular marker for blast-resistant genes that are introgressed in the recurrent genome, in our recurrent genome, which is the MR219, and the donor genome is from Pongsu 1001, the source of blast-resistant gene. They were able, this team were able to produce Padi U Putra 1, which has resistance against blast. And this whole project, I believe, only takes until back cross to F2 generation. So that is a vast difference between the conventional approach versus with the using marker-assisted breeding. These are some the characteristic of Padi U Putra 1. Next. Concurrently, there are also this project team also produced Padi U Putra 2, a submergent tolerance variety, which is very important because, as I already mentioned, flooding is also a big risk that happen in Malaysia every monsoon. Yeah, people complain about huge yield loss due to flood. So Padi U Putra 2, a submergent tolerance variety, also produced via marker-assisted back crossing technique. Now the donor parents is from Swarna Sub 1, which is a variety which has submergent tolerant, and it's the gene, the submergent tolerant gene, is introgressed into MR219 variety. So it has a higher parent genome recovery rate, also only taken 2 generations of back crossing, and then selection of lines that are produced from this indicate that high parent genome recovery. So the performance of these back cross lines are similar to the recurrent genome, which is a high yielding variety what with the addition of the submergent tolerant genes. It also has high yield and submergent tolerance to flooding. Yeah. Next. Just still looking at the first strategy in the trust strategic 1, exploring. We are also looking at exploiting germplasm for tolerance gene. In this research, we are looking at different Malaysian traditional rice accession as germplasm source of abiotic genes, abiotic stress tolerance genes. Here we are looking at 15 traditional rice varieties to look for drought tolerant genes in this traditional rice. So we expose them to drought versus control. Next. Based on this correlation, we identify the drought indices that indicate that have high correlation with high yield under stress. So we have identified these indices to be to have high correlation with yield. And next, by looking at those that score the highest for those indices, we have identified several traditional rice genotypes with potential genes to be exploited as the source of drought-tolerant genes. So these varieties, Babiliong, Banjang, and Padi Darawal, those are all traditional varieties, and we can use these varieties as donor parent in future breeding program in the development of drought-tolerant rice. Next. This graph is just showing some of the performance of these 3 varieties that we have identified, the traditional varieties. As the harvest index, you can see that the performance of Babiliong, you can see the arrows, Banjang, and also Padi Darawal. The performance in under drought treatment and versus the control treatments are not far apart. And looking at the photosynthetic rate as well, during drought and then after drought, you can see that Babiliong, Banjang, and Padi Darawal has resilience, so they recover after drought imposition if is lifted. And you can see that the photosynthetic rate for these varieties becomes higher to recuperate from the stress that they were experiencing under drought stress. Next. We also explore alternative food and feed under the strategy of number 2 to identify alternative food and feed. So we are looking at development of forage and grain maize. We select based on phenotypic and we identify QTLs related to yield and quality for development of maize for animal feed. And then we also explore on alternative food and feed resources by looking at potential crop, sorghum by color, which are known as its climate resilience, drought resistance, and also ability to grow in marginal areas. And it is also found to have high nutritional value for human and animal consumption. So we know that we rely on maize a lot for animal feed. We imported almost 100% of maize for animal feed. Next. For our poultry industry. Next. But we can also use maize as forage crop as it has high protein and good palatability. But of course, in tropical country, we usually use grasses and napier. But we can also propose maize as potential forage crop as it has a better digestibility and palatability compared to other pastures. Next. So this is done in our research, trying to identify genome chromosomal regions that are responsible for traits that are good for biomass yield and plant height. Next, as well as protein content for the quality and fiber content. So when we identify this genomic region, we can use these markers as our selection tools in developing new varieties for maize for animal feed. Next. So these are the potential application we can select using the molecular markers, and then also focus on important traits for phenotypic selection in developing maize variety. And we also look at sorghum, as I said, it's a potential feed and for feed as well, and it has no gluten. So this is a positive quality of sorghum as an alternative for those who are allergic to gluten, which is available in wheat-based products. Recently we produce these new varieties for animal feed, which is potential to be used as animal ruminant industry. Next. The sorghum breeding program has gone. This is just to show some evaluation that we are doing, comparing in many agronomic inputs, and we can see that the performance of sorghum with and without nitrogen. Next. The yield, with in blue and without nitrogen in orange, and also the crude protein content is higher compared to. So the range of sorghum crude protein content is around 9 to 15 compared to Napier grass, which the CP is around 9.79. So it provides a potential alternative for animal feed industry. Next. I will just conclude very shortly the future direction, applying artificial intelligence in breeding to enhance predictive analytics, to enable us to identify the optimum breeding strategy and accelerate development, as well as advance in genomics can help us to increase the precision in selection as well. Ultimately producing higher yield, disease resistance, and adaptable to changing climates in crops. These are the grants that we obtained that are given to UPM to produce all of the breeding programs. And also thank you to collaborators, PG, undergrad and postgraduate plant breeding students. And I think that's all for my talk. I already exceeded the time given. Thank you so much for listening. Assalamualaikum.
All right, thank you very much, Dr. Shima. Please take a seat. Question and answer session will be pooled. We will move on to the next speaker. Our next speaker is Miss Emilia Grace Kasianus. She's a plant breeder from Enza Zadin Asia R&D Sendirian Berhad. She has a lot of experience in cucumber breeding, and today she will be sharing with us about how advanced breeding technologies can drive both speed and precision in variety development. With that, I will hand over to Ms. Emilia.
Thank you, Prof. Uma, for the nice introduction. Yeah, so my name is Emilia. I'm a breeder from NZZ in Asia. And I will talk a little bit about the advancement of breeding technologies for seed enhancement. So this is the outline of my presentation. I will talk a little bit on the challenges in agriculture, advanced technologies to breed for resilient crops, something we just heard from Dr. Shima just now, and the strategic way of also integrating those advanced technologies. Yeah, correct. Yeah, so this is the value chain of the seed vegetable industry. I will talk a little bit on that. So typically, the vegetable value chain industry is driven by market pull. So that starts from the downstream of the value chain from consumer, then to mid segment of the value chain, and then eventually to the seed company, whereby the seed company then will fulfill the needs of the market demands. So for example, the consumer, they want better eating quality vegetables. And then the retailer and the trader, they want a better shelf life. And then the grower, they want high-yielding varieties and also pest and disease-resistant varieties. So then seed company, the breeding company, will try then to breed for varieties that fulfills the demand of the market. But this also goes in complement with technology push, whereby new innovative trait comes from the seed company. And then this trait is not yet in the market and it's not yet demanded by the market. So seed company will then try to find for market opportunities and to introduce this product model into the market. So they create promotional activities like educating the market or demonstration, field demonstration, in order to reshape the customer's preference. So in concept, commercial breeders, we focus on a very different set of traits that can fulfill the demands of the stakeholders. So what you see here is a wholesale market from all over Southeast Asia. We have hot peppers and pumpkins from Thailand. And then we have also cucumbers, a little bit of gourds in Vietnam, and then shallots and gourds also in the Philippines. So while this is these nice, colorful produce, they are the backbone of the farming communities, yes, but this is also shaped by the traders and consumers' demand. But behind these colorful photos, colorful produce is a web of complex challenges. For example, rising temperatures. What you see there on your left-hand side is a fully grown cucumber, but is under intense heat stress in Thailand. So it suffers from tip burn and it's not growing anymore, and it produces a lot of unmarketable fruits. And then what you see on the second photo is an aerial photo of farmland that was destroyed by Typhoon Karding in the Philippines in just a single event. Then we have also evolving pest and disease dynamics. And here we have a virus-infected bitter gourd, also a fully grown plant, but heavily infected by the virus. And then we have fruit borer outbreak in Malaysia that causes economical damage. And then here, this is a photo of a very laborious and resource-intensive method of crop protection in Sri Lanka. The growers, they need to cover individual plants because there isn't any variety that is resistant enough to sustain the high virus pressure in the field. So yes, this is a map of from Carbon Brief. It maps the extreme weather events that impacts crop production all over the world. So what you see in Southeast Asia, the crops are mostly suffering from drought and heat. And then in Europe, it's pretty much everything from drought and heat to extreme cold and winter as well. And then we see the same in Africa, also in North and South America. It's a mix of extreme weather events. Then what you see on your right is the crop impacted by these extreme weather events from 2023 to 2024. So the biggest, combined with the market dynamic shift that is rapidly evolving, seed company needs to be more responsive and more resilient than ever. So we need to breed for varieties that are heat tolerant, varieties that are drought tolerant, varieties that are flooding tolerant, for example. But at the same time, these varieties needs to also be resistant against pests and diseases. These varieties also needs to have reliable yield across all seasons. And to safeguard future adoption, we need to have also conservation of genetic diversity. And how do we deliver this to the market is through seeds. So seed is the delivery vehicle for genetic improvement. But as we know, the conventional breeding, it is slow. It is no longer sufficient to keep up with the pace of the market demands and the agricultural traits because it is limited by longer generation cycles, slower breeding progress, and use of a lot of resources like land, labor, and time. To breed for crops that are resilient fast, we need to have first speed, and it goes hand in hand with accuracy. So what do we mean by speed in breeding? It is the reduced time required for cultivar development and commercialization. So what's the key for us to accelerate breeding progress is first we need to increase the number of generations per year, because some crops, they take 4 to 8 years, sometimes 12 years to be introduced into the market. So this is slow. And for seed company to introduce new varieties, improved varieties into the market faster, we need to also accelerate genetic gain and line fixation. And this genetic gain that is driven mostly by phenotyping, careful selections from the breeder. And if we are to increase the genetic gain in our varieties, this phenotyping from the breeders needs to also keep up. And then when we talk about accuracy, like I said, it needs to go hand in hand with speed. We need to have speed, yes, but we should not compromise on the precision of the selection that the breeders are doing. So 2 key aspects in accuracy of plant breeding is quality phenotypic data and quality genotypic data as well. And these 2 data sets, when they are reliable, breeders can make smarter and faster decisions. Right, so moving on to modern breeding tools. We need nowadays modern breeding tools. We cannot rely only on conventional breeding to introduce speed and accuracy. And one of our powerful tools is controlled environments. So by regulating light, temperature, humidity, and other growing conditions, we can have multiple generations per year. And that will also accelerate line fixation. And if an environment is tailored to a specific breeding goal, it can also help breeders. It will reduce the noise, unwanted noise coming from the environment, so that will help breeders to make effective selections. And these effective selections, it goes hand in hand with the genotype, so that we understand better the QTLs or the targeted genes that we are phenotyping on. And when we are talking about genomics, one important tool is GWAS, that is Genome-Wide Association Study. So GWAS is the study of a targeted trait or the desired trait across a wide range of genomes. So when we, in other words, it's gene mining. So when we talk about diverse backgrounds of genetics, it does not need only to come from the breeding lines, elite breeding lines. It can also come from land races, wild accessions, for example. And the key to gene mining is to have a correct selection pressure. So for example, if you are selecting for powdery mildew resistant, you need to expose your materials to high powdery mildew field. And then if you are selecting for heat tolerant, for example, so you need to expose your accessions in a very heat stress condition as well. So take, for example, here, these are pumpkins that are screened in Thailand under a very intense heat. So we see here plants that are susceptible to heat. They are completely dried up. They produce very little to no fruits. And we have plants who have medium vegetative vigor, just enough foliage to sustain some fruit bearings, and just enough leaves for the plants to also allow fruit maturity to be harvested. And then we have some outstanding plant here that is very vegetatively vigor, but also with nice fruit settings, despite being subjected to very high heat condition. So these kinds of variations, that is really important to be recognized by the breeders in the field, which is why quality phenotyping is also part of gene mining, and that will be associated with the genotypic data. So the key is to subject your accessions to appropriate heat condition in order to reveal genetic differences. Yeah, so as I was saying, gene mining also studies genes across wide, diverse genetic backgrounds. So hot peppers and eggplants, that is a representation of diverse genetics. And here we have the population tree structure that visualizes how different accessions are genetically related to each other or how different they are from each other. So in other way, GWAS, because it can also detect novel alleles that are already lost due to domestication. In a direct way, GWAS can also introduce some diversity into your breeding program. Yes, so when we have the targeted gene, for example, from GWAS, gene mining, we can then introgress this trait of interest via marker-assisted selection. So when we integrate GWAS and marker-assisted selection together, it can facilitate gene gain, genetic gain, not only single trait at a time, but multiple traits at a time at a shorter breeding cycles. And that can further accelerated by automation in the marker lab. So here we have a marker lab in NZZ. It is fully automated from leaf sampling until marker runs. So with this automated marker lab, we are able to run 73 millions of markers every year. And then when we talk about gene editing, that is also a tool that is getting popular each day, and it allows for an approach such as gene silencing of susceptible genes and also gene knockout. So for example, in barley, the powdery mildew resistance is introduced by the knockout of MLO genes. So no matter how advanced your how advanced the genomic tools or how efficient your genetic genomics tools has become, it does not mean anything without quality phenotyping. And phenotyping is a very laborious work, and as I was saying, phenotyping is also one way we can speed up product introduction into the market by rapid phenotyping. But that is now facilitated today by digital phenotyping. So take, for example, that is a machine of fruit phenotyping in NZZ. What we see is perfectly clustered tomatoes, but the machine is able to extend its phenotype beyond just that. It is able to measure uniformity. It is also able to measure the defects that is happening internally. So if this kind of machine is also designed for the other crops, for example, in the lower photo, that is bitter gourd that is cut into half, and then what you see inside is the defects caused by fruit fly. So with this kind of machine, then we will save a lot of time without having to cut the fruits. We know what's happening inside that is caused by the fruit flies. And then we have digital phenotyping of lettuce in NZZ's open field, for example. So yeah, so I've explained now about controlled environments, about genomes, GWAS, marker-assisted selection, and gene editing, and then also phenotyping through high-throughput phenotyping. The idea is not to use these tools in isolation of each other. To be competitive in the market, to introduce product with speed and accuracy in the market, we need to integrate these tools together, so that we can make a smarter and faster decision. And that is also the basis of predictive breeding, which is also an AI-based breeding. Finally. Yeah, plant breeding is a multidisciplinary work, yes. So despite having all these machines, like the phenotypic machines, the breeder, they still need to be in the field to do the phenotypic on the plants. The machine, they do not phenotype everything. And then you have, in the genomics field, we still need the biotech breeder. They are the experts in the genomics field. And in enviromix, we have the operational team who is an expert that tailors for the growth condition of the plant. So it is a joint effort between different experts from different fields, this plant breeding. It's not a single effort by the plant breeder itself. So, thank you.
Thank you very much, Ms. Emilia.
Yeah.
You may take a seat. We will have the Q&A later. The next presentation is by Dr. Muhammad Shaifuddin. This paper, sebenarnya kertas Dr. Muhammad Syaifuddin sepatutnya pada hari pertama ya. Tetapi ada sedikit perubahan, jadi kita terpaksa bawa ke hari ini dan kertas adalah dalam pembentangan dalam bahasa Melayu. Okey. Tapi ini pembentangan saya rasa paling penting untuk simposium kita sebab dia adalah hala tuju benih negara. Apa strategi untuk hala tuju benih negara? So tajuk pembentangan beliau adalah hub benih negara, langkah strategik memperkasa industri benih tanaman tempatan. Please allow me to introduce the speaker. Dr. Muhammad Syaifuddin lahir di Kelantan. Sekarang merupakan pengarah Pusat Perancangan Strategik dan Pengurusan Inovasi MARDI. Kepakaran beliau adalah Sensors and Precision Farming. Pernah memegang pelbagai posisi di MARDI, ya, pengarah banyak tempat, Pusat Komunikasi Korporat MARDI, pengarah Pusat Penyelidikan Padi dan Beras Negara dan sekarang bukan negara MARDI, dan sekarang pengarah Pusat Perancangan Strategik dan Pengurusan Inovasi MARDI. Right, so dengan ini saya mempersilakan Dr. Muhammad Syaifuddin untuk memberikan pembentangan. Terima kasih.
Terima kasih Prof. Umar. Assalamualaikum warahmatullahi taala wabarakatuh dan selamat petang. So saya nampak Malaysian dalam ni, okeylah. Pembentangan saya dalam bahasa Melayu. Okey, hak benih negara. Kenapa pentingnya hak benih negara? Kita dah ada National Gene Bank di MARDI. Kita ada pusat benih yang baru ditubuhkan di UPM, Pusat Benih Negara. Kita ada pelbagai lagi lah hub-hub yang berada dalam negara. Tapi di peringkat kementerian, mereka merasakan perlu ada pengembelingan untuk hub benih ini supaya dari segi peruntukan, dari segi strategi plan tindakan perlu disasarkan secara pengembelingan, iaitu kesatuan di antara sama ada di Jabatan Kerajaan ataupun di peringkat swasta, di peringkat universiti dan sebagainya. Jadi di peringkat KPKM telah mengarahkan MARDI untuk membentangkan satu kertas dasar sebenarnya untuk hak benih negara ni. Okey, next. Okey, kalau tengok kepada senario sekarang, kenapa perlunya benih yang berkualiti? Satunya adalah kita nak menghasilkan atau mengeluarkan tanaman yang berkualiti. Daripada tanaman yang berkualiti, kita dapatkan hasil makanan yang berkualiti untuk keterjaminan makanan negara. Dan kalau kita tengok senario yang berlaku sekarang ni, 95% daripada benih sayuran terutamanya kita import daripada luar negara. Dan kalau tengok daripada senario padi, padi ni kita dapat kawal lah. Dan kalau tengok dari segi pencapaian MARDI, kita telah pun menghasilkan 58 variety dan hampir 95% padi yang bertanam dalam negara adalah menggunakan variety MARDI lah. Terima kasih kepada UPM telah mengeluarkan Putera 1 dan Kambing Putera 2. UKM pun ada UKM RC 1 dan 2 dan IGC Nuklear dia mengeluarkan IS21. Dan kalau kita tengok dalam rancangan Malaysia ke-12, MARDI telah pun diberikan peruntukan yang tertentu hasil kajian daripada MARDI. Dan kita telah pun membuat satu perancangan rapi untuk meneruskan aktiviti ini dalam rancangan Malaysia ke-13. Next please. Okey, ini adalah kerangka ataupun model hak benih negara yang kami sepatutnya bentang kepada Majlis Benih Negara pada 2 minggu lepas. Tapi disebabkan oleh YBM yang kesibukan ke Rusia dan akan ke Jepun, ditangguhkan dalam masa tertentu lah. Dan ada 3 objektif utama, 5 strategi, 30 plan tindakan dan 5 impact. Dan kalau kita tengok 3 objektif utama adalah yang pertamanya adalah untuk mengukuhkan sumber benih dan baka tempatan. Yang kedua adalah memperkasakan pengeluaran benih dan baka tempatan. Dan yang ketiganya adalah meningkatkan akses kepada benih dan baka tempatan. Next please. Okey, untuk hak benih negara ni, sekarang ni ada 3 segmen utama lah. Iaitu dari segi sektor tanaman yang melibatkan MARDI dan Jabatan Pertanian. Ini saya melihat membentangkan dari segi kerangka KPKM. Dan kita ada sektor ternakan di bawah MADI dan juga DVS, dan juga sektor perikanan di bawah Jabatan Perikanan Malaysia. Okey, untuk 5 strategi hak benih. Next. Kita ada 3 objektif utama yang seperti yang saya semaklukkan tadi, iaitu pengukuran sumber, pemberkasan pengeluaran dan juga peningkatan akses. Di dalam pengukuran sumber kita ada 2 strategi utama, iaitu pemulihan sumber genetik tanaman dan juga pengukuran program pembaikbakan tanaman. Di bawah pemberkasan pengeluaran kita ada 1 strategi, iaitu meningkatkan kapasiti pengeluaran benih asas dan juga mata tunas tempatan. Dan di bawah objektif ketiga kita ada 2 strategi, iaitu pemberkasan usahawan pengeluaran benih dan bahan tanaman dan juga hak benih sebagai pemangkin rantaian industri benih. Next please. Okey, ini adalah pemeliharaan sumber genetik tanaman. Seperti saya sebut tadi, kenapa kita perlu preserve sumber genetik ni. Satunya adalah untuk kita memastikan kita dapat mengeluarkan benih-benih tanaman tempatan yang berhasil tinggi, berdaya tahan, iaitu tahan penyakit, tahan kepada perubahan cuaca dan sebagainya, dan juga ada sesuai untuk perubahan persekitaran tempatan. Itu antara yang penting. Dan di MADI kita ada bank jeng kemasaan yang telah ditubuhkan pada tahun dia bermula pada tahun 74, dan kemudian jeng padi di MADI Subang Perai telah pun ditubuhkan pada tahun 1989, dan kita diwartakan sebagai bank jeng kemasaan pada 2014. Dah pun lebih daripada 30 tahun lah kita telah berada dalam industri. Dan kalau kita tengok Janas Pasma MADI, kita ada 9 lokasi di setiap negeri di seluruh Malaysia. Dan ini adalah ladang Janas Pasma benih-benih tempatan, termasuklah benih-benih keluaran MADI dan juga yang MADI ada dalam Jim Bank kemasaan. Dan di bawah DOA dia ada petak repository dan juga arboretum yang menghasilkan 290 varieti tanaman dan 1,400 bilangan pokok sumber di 18 pusat dan lokasi. Ini adalah di bawah kelolaan DOA. Next please. Okey, untuk pengukuhan program pembaikbakan tanaman, kita ada beberapa komponen, iaitu yang pertama adalah breeding, seperti yang disebutkan oleh Profesor tadi dan juga Miss Emma. Kita ada pemilihan berasaskan prestasi, iaitu selection. Kita ada pendekatan bioteknologi, gene editing dan benih tissue culture. Ini adalah topik-topik ataupun pengkajian di MADI yang telah dihasilkan dan yang recently kita embark dalam gene editing. Dan kalau kita tengok 5 dekad menabur bakti, MADI telah pun menghasilkan 56 dan juga 53 klon untuk tanaman industri, 82 klon untuk horticulture dan juga 4 baka untuk ternakan. Ini adalah varieti-varieti yang dihasilkan oleh MADI. Dan kalau kita tengok baru-baru ini kita telah melancarkan cili L5, cili MADI dan juga varieti bawang MADI. Okay, next please. Okay, untuk peningkatan kapasiti pengeluaran benih asas dan mata tunas tempatan, kita ada kebun benih mata tunas buah-buahan di MADI Sedang, pengeluaran anak benih tembika di MADI Bacok, kebun benih tisu kaca pisang di MADI Kelang, dan untuk padi kita mengeluarkan benih baka dan benih asas. Benih daftar dan juga benih sah dikeluarkan oleh syarikat-syarikat yang berdaftar di bawah KPKM. Dan untuk pengeluaran benih baka dan benih asas padi MADI, ia memenuhi ISO lah. Kami ada ISO di bawah ISO 9000. Next please. Okey, untuk pemangkasaan usahawan pengeluaran benih dan bahan tanaman, seperti saya masukkan tadi, kita ada pengembelingan dan pertukaran yang telah diberikan, dan kita telah pun membangunkan usahawan-usahawan tertentu untuk benih-benih MADI. Dan kita telah pun mengeluarkan beberapa usahawan, iaitu untuk cili, pisang, buah-buahan, kopi, bawang merah dan juga kacang tanah. Dan ini adalah yang recently lah yang kita mengeluarkan benih bawang dengan kerjasama beberapa usahawan di Perak, di Kelantan dan juga di Lembah Kelang. Okay, next please. Okey, untuk strategi kelima adalah hak benih sebagai pemangkit rantaian nilai industri benih. Ini adalah kerjasama yang telah kita adakan sama ada dalam negara dan juga institusi di luar negara. Okey, next please. Untuk perkembangan terkini untuk hak benih tanaman, kita telah pun membuat sesi libat urus dan satu pembentangan di Jawatankuasa Kerja Makanan, satu canvas projek mega di bawah RMK 13 dan kita pun juga telah pun memohon pertukaran RMK 13 untuk RP1 ni khusus untuk hak benih tanaman dan kita seperti yang saya sebut tadi kita diminta untuk mengadakan satu sesi pembentang Jun nanti. Next please. Okey, saya dah habis. Cuma saya nak promote sedikit. Di Madi akan ada Showtech Madi 2025 yang akan berlangsung pada 29 Mei hingga 2 Jun 10 pagi dan 10 malam memaparkan teknologi Madi, usahawan-usahawan bima Madi dan lain-lain lah. Saya menjemput semua yang berada dalam dewan ni dan untuk sama-sama datang di Showtech Madi 2025. Masuk adalah percuma, parking adalah percuma di MAIF dan di sekitar Madi Ibu Pejabat. Next please. Sekian, terima kasih.
Okay, thank you very much Dr. Muhammad Saifuddin for that very interesting talk. Kita nampak mungkin tak banyak info di situ kerana masih perlu dibentangkan di peringkat yang lebih tinggi dahulu ya. Tapi sekurang-kurangnya kita dapat tahu apa strategi hub benih negara. Okay, so sekarang saya ingin membuka kepada Q&A. U Putra rice is claimed to be high yielding and disease resistant. Why this variety is not widely adopted in major granary areas such as the IADA Barat Laut? Silakan Dr. Shima.
For you, I understand the breeding trials are done in granary Barat Laut is involved or not in the trials as well. So now it's already available. I think the seeds are already available and produced by companies, seed companies. I think it's readily available to be adapted and adopted lah by farmers to plants in their plots. The second question, who produces the seed for U Putra? Also, I think this they have seeds companies, Haji Makno, yeah. They are registered companies that produce benih berdaftar dan benih besar. Benih sah dan benih daftar. So the benih bakar tu by UPM, but for benih daftar and benih sah for commercial purposes, these are produced by Haji Makno Company. Yeah. For you, Padi Putra.
All right, thank you, Dr. Shima. Any more questions from the audience, either physically or you can type? Dah letih ke? Soalan? No? No more questions? Okay, yes, please.
Assalamualaikum warahmatullahi wabarakatuh. Saya Nur Asmi Musa. Saya nak tanya macam mana nak dapatkan benih-benih yang dikeluarkan oleh pihak Madi untuk diusahakan oleh pihak industri.
Okay, obviously.
Boleh saya tahu benih apa tu? Benih sayuran ke, benih buah-buahan ke, benih padi ke? Okey, benih sayuran buah-buahan lah. Okey, Madi sebenarnya di setiap negeri kita ada mengeluarkan benih-benih. Dan yang terbesar adalah di Madi Ibu Pejabat lah. Kita ada pusat benih Madi. Dan di situ pelbagai jenis varieti Madi yang dikeluarkan. Dan yang pentingnya benih-benih Madi ni dia ada ISO dia. So maknanya dia berkualiti lah. Kalau tuan di Lembah Kelang, saya cadangkan pergi ke pusat benih madi. Kat sini pun kita ada wakil dia. Jadi di negeri-negeri kita ada. Di negeri-negeri Pantai Timur, Utara dan sebagainya memang ada lah. Cuma dia berpusat ikut lokaliti. Contohnya kalau mungkin di Kuala Kangsar, dia banyak kepada durian. Sebab di situ adalah durian tourism. Kalau di Johor contohnya di Keluang kita ada benih kopi. Sebab di situ adalah pengeluar kopi. So kalau di HQ ni semua adalah termasuklah durian, buah-buahan, sayuran dan sebagainya. Benih variety madi dan juga ada benih-benih lain yang dalam koleksi kami lah. Boleh datang. Kat sini pun ada saya rasa. Kalau nak kuantiti besar kena datang bincang lah.
Booth Mardi ada di luar Ada booth Mardi.
Ya.
Ya. So mungkin boleh melawat booth lah untuk maklumat lanjut. Ada soalan lagi? Daripada peserta ataupun daripada Slido tadi Adakah, bilakah variety baru padi akan dikeluarkan oleh Mardi?
Kita ada 2 lagi variety yang akan keluar pada tahun ni. Satu adalah MR333 yang berhasil tinggi. Dia akan menggantikan MR297 lah. MR297 ni hampir 50% ditanam dalam kawasan bertanam lah, especially Jelapang. Bulan Ogos adalah untuk MR33 dan kita ada satu lagi CL3 dengan CL4 yang kami bekerjasama dengan BASF akan keluar bulan Julai nanti. Yang itu adalah untuk menangani masalah padi angin.
Thank you. Ada soalan lagi?
Assalamualaikum. Ya. Okey, saya nak tanya pada Dr. Sharifuddin sebab strategi nombor 2 tadi ada 5 strategi. Salah satunya benih tissue culture. Mana keberkesanan benih TC ini kepada industri nanas negara? Sebab setahu saya benih tak cukup dan permintaan world demand market for the nanas adalah amat lumayan lah.
Okey, untuk sekarang ni yang paling besar yang kita keluarkan benih kita ni semua ada ISO lah. Yang setahu saya memang ada dalam pipeline kita, dalam mungkin Puan Zeti kita dah keluarkan ke? Sebab Puan Zeti di bawah benih. Mungkin saya minta dia terus jawab lah.
Saya tambah sikit lah berkaitan dengan sukan nenas. Saya dimaklumkan memang dari segi dasar lembaga nenas kita memang bekalan bahan tanaman tissue culture masih belum dibenarkan kan. Tapi dari segi studi yang dibuat oleh pakar kami di Pusat Bioteknologi memang kita dah tengok dari segi sumber tissue culture pisang nenas dan kita dah buat tengok performance dia di ladang. Memang hasil dia kualiti dia sama macam maksudnya dia punya hasil tanaman tak menjejaskan Zuraida lah saya sebutlah juga sebab benda ni ada dibentangkan juga dalam jawatankuasa di bawah jabatan lah ada jawatankuasa pengesahan bahan tanaman dan memang dimaklumkan yang itulah cuma dari segi R&D saya boleh kata teknologi itu tak ada masalah untuk SENAS sebenarnya. R&D saya boleh kata teknologi itu tak ada masalah untuk SENAS sebenarnya. Yes. Okay.
Puan
Zaiti, apakah variety kacang tanah yang telah dikeluarkan?
Yang ada sekarang adalah Magenta. Dia adalah salah satu yang pernah dikeluarkan tahun yang Madi adalah yang terkini. Dan kalau siapa nak bahan tanaman boleh hubungi Madi Bacok lah.
ataupun pada saya boleh contact lah kalau perlukan bahan tanaman tersebut.
Tidak ada soalan, saya ucapkan pengacara majlis, Dr. Syazwan.
Thank you very much to Professor Dr. Umarani for moderating the session and as well as to all of the distinguished speakers. Ladies and gentlemen, now I would like to invite Puan Nur, the token of appreciation on behalf of the organizing committee. Now let us invite Yang Berbahagia Professor Dr. Umarani Senior, President of National S.E.E.D Association Malaysia, as the moderator of the session. Okay, Prof, please remain on the stage as usual. We're going to take picture after this. All right. Next, Dr. Nazatul Shima binti Naharuddin from University Putra Malaysia. Let us invite Miss Emilia Grace Cassianus from Enza Zadain Asia R&D Sendirian Berhad. And last but not least, Dr. Muhammad Shaifuddin bin Abdul Rahman from Malaysia Research and Development Institute, Mardi. Right, can I have everyone in the center of the stage for a group photo? Is it okay? Tengah sikit.
you.